And now we are going to start with our last Ignite talk before the debate. So we have 3 gentlemen this time. We have 2 microphones. And it seems like this is going to be an interesting one. Yep. You have a slide changer.
If any of you watched TV as kids, you're probably familiar with the Charmin Bear commercials. Remember, best for bare bottoms? Well, the young Charmin Bear from our childhood, Carmen, has grown up and is doing some soul searching. After spending her life in the advertising industry with her mother, she feels a burning desire to meet her father. She has come to us, the biologists of BioG 1500, to discover the identity of her father once and for all. She's narrowed it down to 2 options. Either Bruno, the Brown University bear, or Touchdown, the big red bear. So, can we help Carmen find her father's identity using microsatellite DNA? First questions first. What is microsatellite DNA? Microsatellite DNA, also known as short tandem repeats, or STRs in the forensics community, are repetitive DNA sequences that can be found in our genome. When DNA replicates, sometimes the newly synthesized complementary strand can actually slip backwards, causing the DNA polymerase to synthesize an additional copy of the slipped sequence. Proofreading mechanisms fix most errors caused by slippage, but in certain non-coding regions of the genome, the repeated sequences can remain without consequence. Repeated sequences with 1 to 6 base pair motifs are called microsatellites, or STRs. Some of these STRs can be used as genetic markers to identify species or even individuals. Every individual has 2 copies of each allele, one from the mother and one from the father. So now, going back to Carmen's genome, her TH01 allele has a repeat sequence of TATC at the TH1 allele. Can anyone tell me how we would classify her genotype at this location based on the number of base pair motif repeats? Can anyone count them? 8 and 6, absolutely right. So is she heterozygous or homozygous for this allele? Heterozygous, that is right. So, now we've got to ask the question, just how effective are these STRs at predicting an individual's identity? Or just how unique is Carmen's pattern of microsatellites? We can find the theoretical frequency of a genotype in a given population using the Hardy-Weinberg equilibrium. If we call P the frequency of the 6 allele, and Q the frequency of the 8 allele, then the probability of being homozygous for 6 is P squared, the probability of being homozygous for 8 is Q squared, and the probability of being heterozygous for 6 and 8, like Carmen, is 2PQ. So here we have part of Carmen's STR profile that shows the locus, her genotype at that locus, and then the frequency calculation for her genotype. Because her alleles are sorted independently, we can multiply the frequency at each locus to find her random match probability. The probability that a random bear in the population will have the same alleles as she does at these 4 loci. Her random match probability at these 4 loci is 2.78 times 10 to the negative 11th, which is pretty unique considering we've only taken 4 loci into account. On the other hand, my random match probability was pretty much exactly the same given 10 loci, so she is way more special than I am.
To find out Carmen's genetic profile, a small amount of DNA extracted from her cheek cells was subjected to the polymerase chain reaction, or the PCR, to mass replicate this DNA for analysis. This process allows for the simultaneous amplification of several loci of interest, called multiplexing. This process is fast and cheap, so it is widely used for paternity testing. 4 components are needed to run a PCR. First, Carmen's DNA as template. Second, a pair of short single-stranded primers complementary to each targeted locus. Third, a DNA polymerase called the Taq polymerase to catalyze the formation of new DNA strands. And fourth, 4 deoxynucleotides to serve as raw materials. So, how is the PCR carried out? Well, it's carried out in a cycle with 3 different temperatures, first at 95 degrees Celsius, then 4260, and finally 72. At 95 degrees Celsius, the heat breaks the the heat breaks the hydrogen bonds between the double strands. At the lower 40 to 60 degrees, the hydrogen bonds are restored to allow primers to attach to the template. At 72 degrees, the Taq polymerase catalyzes the formation of new DNA strands. Typically, a PCR is run for about 30 cycles, producing more than 1 million copies in just 3 hours. Now, how are PCR products analyzed? Well, two detection methods are commonly used: gel electrophoresis and electrophoregram. Gel electrophoresis makes use of an electrical field which analyzes the DNA molecule sizes based on the migration distances. The setup includes a positive and a negative terminal, as well as a medium consisting of agarose, which is inert and has equally sized pores through which DNA travels. The phosphate groups on the DNA gives us a negative charge, so it travels from the negative to the positive terminal. Larger molecules travel slower, while smaller ones travel faster, and therefore closer to the positive terminal. The results are then compared to a standard curve with known fragment sizes to determine the number of base pairs of carbon's DNA.
Okay, so we have looking at gels from electrophoresis as one way to help out touchdown, but let's take a look at another method: electropherograms. What is an electropherogram? Well, it's basically a more quantified, more accurate version of gel electrophoresis. The way that it works is that dyes are covalently attached to PCR primers with different colored dyes attached to primers that produce similar length products. That way, we can differentiate alleles from different loci that are similar lengths. The DNA are then analyzed using a machine with capillary tubes that are very sensitive to fragment size. Finally, the data are displayed in an electropherogram. So the way that we read this thing is by looking at the peaks at each locus. The peaks include how many base pairs are at the locus, and this number correlates to a specific allele. 2 peaks denotes 2 alleles, meaning heterozygosity, and 1 peak denotes 1 allele, meaning homozygosity. Therefore, from an electropherogram, we can determine an individual's genotype at a particular locus. Now, if we know touchdown's genotype at a particular locus, then we can determine if he is Carmen Sharman's father or not. How? By looking at the obligate allele. The obligate allele is the allele on the child's locus that is not found on the mother's locus. So, if Carmen Sharman's obligate alleles are found on touchdown's genotype, then touchdown is the father. But you may be wondering that perhaps some other bear in the population wandering around Cornell has the same genetic profile as touchdown, and we could just be getting a false positive due to chance. Well, we can use the paternity index to check our confidence. The probability that touchdown passed the obligate allele to Carmen, divided by the random match probability that Aaron previously talked about, yields the paternity index. The higher the index, the greater the chance that touchdown is the true biological father. Now, wait a minute. Touchdown? Oh, the results are in! Right? These are the results. Based on these 3 loci, do you think touchdown is the father of Carmen Sharman or not? Yes? Touchdown? When it comes to the fatherhood of Carmen Sharman, you are the father! Thank you.